BTEC Applied Science for AAQ. This video is for Unit 1 Biology. In the last video I talked about amino acids. In this video I'm going to talk about the structure of proteins. In my next video I'll talk about a bit more about the functions of proteins. But anyway, the structure of proteins. So starts off simple, gets more and more complicated. There are 4 levels of structure. There's the primary structure, which is just a polypeptide. Talked about that in the last video. Basically a long chain of amino acids. That's the primary structure. There is a secondary structure, and there are 2 possibilities. You can have what we call alpha helix or beta pleated sheets. Talk a bit more about those in a sec. Okay, and that's the secondary structure. There is a tertiary structure, which is a 3D folded structure of a single polypeptide chain formed by interactions of side chains. That's the tertiary. And then the quaternary is when you have multiple polypeptide chains. So the primary structure, lots and lots of amino acids joined together. To be officially a protein, there needs to be at least 50 of them. Yes? So that's your primary protein structure. Alpha helix and beta pleated sheets. So the alpha helix is, well, a helix is like a spiral. Okay, beta pleated sheets, pleated like the pleats on a skirt, like if you get a strip of paper and you fold it and you pull it apart and it's got like a zigzaggy shape. So alpha helix, beta pleated sheets, and what holds them together, what keeps their shape, are hydrogen bonds again. Okay, and the hydrogen bonds are between the H on the NH and the O on the double bonded oxygen. This is our tertiary structure. Tertiary is when it only involves one polypeptide chain, which has become like a globular thing. It's folded in on itself and it's formed a particular shape. The shape of proteins, as we'll see, is very, very important. Yes? So you've got this globular shape, a globular tertiary structure, and what keeps its shape? Well, just about any type of bonding you can think of. There's ionic bonds where opposite charges form a bond. On the diagram you see this salt bridge there. There's lots of hydrogen bonding going on all over the place. There are these things called disulfide bridges. Yes, and this is where you've got 2 sulfur atoms on 2 different parts of the chain bond covalently, and that forms a very, very strong bond. That's a disulfide bond. And then you've got this weak type of bonding called van der Waals or London forces. Very weak bondings due to temporary charge distributions. You'll do this in chemistry, by the way, van der Waals bonds. Basically, at any particular time, the electrons may kind of be over there on one molecule and over there on another molecule, and the positives and negative charges attract each other. Very, very temporary. Okay, so this is our tertiary structure, which is a globular, like a blob, like a football, held together by several types of bond, usually between the R groups on the amino acids in the chains. Now, the quaternary structure, very, very similar, only if you've got at least 2 polypeptide chains, okay? And as I said, it's a 3-dimensional shape, globular shape, and the shape is very, very important. Things like on enzymes, you've got an active site, and the shape of the active site, you know, it's where things fit in. Very, very important, the shape of these proteins. Hemoglobin. Here's an example of a quaternary structure. Yeah, a hemoglobin molecule. and you've got 4 polypeptide chains. You've got 2 alpha, 2 beta subunits, yes, an alpha chain and a beta chain, and they come together and they form this structure. What does hemoglobin do? Well, it's in red blood cells, and what it does is it carries oxygen around the body. Yes, when it's got oxygen on it, then it's oxyhemoglobin, and then it can release its oxygen and just become hemoglobin again. Now, we can classify proteins, we're talking the tertiary and the quaternary ones, into 2 types of structure. There are globular ones, which are like a blob, but then there's also fibrous ones as well, like fibers. The globular ones are compact and spherical and water soluble, and they perform metabolic functions such as enzymes, I've talked about, hormones, antibodies as well. They have very definite shapes on them so that the antigens fit on there. Hemoglobin is a good example of a globular protein. But then we've got fibrous ones as well. They are long, they are insoluble, like strands, strand-like, and they tend to provide strength and support in structural roles, jobs that, keeping the structure of things like in hair and in tendons. A couple of examples: keratin, is in your hair and in your nails, and collagen is in connective tissue like tendons and bone. So you have globular and fibrous, 2 examples of the tertiary or quaternary structure proteins. Okay, so here's some questions. Have a go at them. And again, I'm going to be very lazy. I'm not going to tell you the answers because I already have.